Through evolution, cells have built protein machines that are capable of carrying out most biological reactions. Cells employ protein machines for the same reason that humans have invented mechanical and electronic machines. For almost any task, manipulations that are spatially and temporally coordinated through linked processes are much more efficient than is the sequential use of individual tools. Covalent modification controls the location and assembly of protein machines. Protein machines and other protein complexes play an important role in the life of the cell, but these complexes do not sit around in the cell interior, pre-assembled and poised for action. It has recently become clear that most protein machines form at specific sites in the cell and are activated only when and where they are needed. This mobilization is generally accomplished by the covalent addition of a modifying group to one or more specific amino acid chains on the participating proteins. More than 200 types of covalent modifications can occur in the cell, each helping to regulate protein function. We have so far focused on the addition and removal of phosphate groups. As we saw earlier, phosphorylation can increase or decrease a protein's activity. See Figure 438. But it can also promote the assembly of proteins into larger complexes. For example, extracellular signals can cause the phosphorylation of a class of transmembrane proteins called receptor tyrosine kinases. This phosphorylation often triggers the assembly and activation of elaborate intracellular complexes of signaling proteins, which pass along the message to grow or divide. See Figure 1630. Other covalent modifications have different regulatory roles. Addition of the fatty acid palmitate to a cysteine residue drives a protein to associate with cell membranes. Attachment of ubiquitin, a 76 amino acid polypeptide, can target a protein for degradation, as we shall see in Chapter 7. Each of these groups is enzymatically added or removed depending on the needs of the cell. A large number of proteins are now known to be modified on more than one amino acid side chain. The p53 protein, which plays a central part in controlling how a cell responds to DNA damage, can be modified at 20 different sites. Figure 444a. Because an enormous number of combinations of these 20 modifications are possible, the protein's behavior can in principle be altered in a huge number of ways. The set of covalent modifications that a protein contains at any moment constitutes an important combinatorial regulatory protein code. The attachment or removal of these modifying groups controls the behavior of a protein, changing its activity or stability, its binding partners, or its location inside the cell. See Figure 444. This regulatory code enables the cell to make optimal use of its proteins, and it allows the cell to respond rapidly to changes in its condition or environment. How proteins are studied. Understanding how a particular protein functions requires detailed structural and biochemical analyses, both of which require large amounts of pure protein. But isolating a single type of protein from the thousands of other proteins present in a cell is a formidable task. For many years, proteins had to be purified directly from the source, the tissues in which they are most plentiful. That approach was inconvenient, entailing, for example, early morning trips to the slaughterhouse. More importantly, the complexity of intact tissues and organs is a major disadvantage when trying to purify particular molecules, because a long series of chromatography steps is generally required. These procedures not only take weeks to perform, but they also yield only a few milligrams of pure protein. Nowadays, proteins are most often isolated from cells that are grown in a laboratory. Often, these cells have been tricked into making large quantities of a given protein using the genetic engineering techniques that we will describe in Chapter 10. Such cells generally allow large amounts of pure protein to be obtained in only a few days. In this section, and in panels 4.4 to 4.6, pages 164 to 167, we outline how cells can be grown in culture and how proteins are purified from these and other cells. In the How We Know section, pages 158 to 160, we describe how these proteins are analyzed to determine their amino acid sequence and their three-dimensional structure. Finally, we shall discuss how efforts to probe protein structure and function are being conducted on a large scale with the hope of obtaining a deeper understanding of how sets of proteins cooperate to make life possible. Cells can be grown in a culture dish. Given the appropriate surroundings, most plant and animal cells will live, proliferate, and even express specialized properties in a tissue culture dish. Experiments performed using cultured cells are sometimes said to be carried out in vitro, literally in glass, to contrast them with experiments on intact organisms, which are said to be carried out in vivo, literally in the living. These terms can be confusing, however, because they are often used in a very different sense by biochemists. In the biochemistry laboratory, in vitro refers to reactions carried out in a test tube in the absence of cells, whereas in vivo refers to any reaction taking place inside a living cell, even cells that are growing in culture. Although not true for all types of cells, most cells grown in culture display the differentiated properties appropriate to their origin. Fibroblasts, the precursor cells that give rise to connective tissue, continue to secrete collagen. Cells derived from embryonic skeletal muscle fuse to form muscle fibers that contract spontaneously in the culture dish. Nerve cells extend axons that are electrically excitable and make synapses with other nerve cells. And epithelial cells form extensive sheets with many of the properties of an intact epithelium. Figure 448. Because these phenomena occur in culture in a controlled environment, they are accessible to study in ways that are often not possible in intact tissues. For example, cultured cells can be exposed to hormones or growth factors, and the effects that these molecules have on the shape or behavior of the cells, or on the proteins they produce in response, can be easily explored. Cultured cells can also provide a ready source of raw materials for biologists interested in purifying and studying a particular protein or protein machine, as we see next. Purification techniques allow homogeneous protein preparations to be obtained from cell homogenates. Whether starting with a fibroblast culture, a piece of liver, or a vat of cells that have been engineered to produce the protein of interest, the first step in any purification procedure is to break open the cells to release their contents. The resulting slurry is called a cell homogenate. This physical disruption is followed by an initial fractionation procedure to separate out the class of molecules of interest. For example, all the soluble proteins in the cell. Panel 4.4, pages 164 to 165. With this collection of proteins in hand, the job is then to isolate the desired protein. The standard approach involves purifying the protein through a series of chromatography steps, which separates the individual components of a complex mixture into different portions or fractions. After each such step, one uses some sort of assay, for example, a test for the protein's activity, to determine which fractions contain the protein of interest. Such fractions are then subjected to additional chromatography steps until the desired protein is obtained in pure form. The most popular forms of protein chromatography separate polypeptides on the basis of their size, their charge, or their ability to bind to a particular chemical group. Panel 4.5, page 166. If antibodies that recognize a particular protein are available, they can be used to help extract that protein from a mixture. See panel 4.3, pages 144 to 145. A similar approach can be used to isolate those proteins that interact physically with the protein being studied. In this case, the purified protein of interest is attached to the matrix of the chromatography column. The proteins that bind to this protein will collect in the column and can then be eluted by changing the composition of the washing solution. Figure 449. Proteins can also be separated by electrophoresis. In this technique, a mixture of proteins is loaded onto a polymer gel and subjected to an electric field. The polypeptides will then migrate through the gel at different speeds depending on their size and net charge. Panel 4.6, page 167. If too many proteins are present in a sample, or if the proteins are very similar in their migration rate, they can be resolved further using two-dimensional gel electrophoresis. See panel 4.6. These electrophoretic approaches yield a number of bands or spots that can be visualized by staining, each one containing a different protein. Electrophoresis and chromatography, each developed more than 50 years ago, have been instrumental in building an understanding of what proteins look like and how they behave. Table 4.2. Both techniques are still used very frequently in laboratories. Once a protein has been obtained in pure form, it can be used in biochemical assays to study the details of its activity, and it can be subjected to techniques that reveal its amino acid sequence and precise three-dimensional structure. See how we know, pages 158 to 160. Large amounts of almost any protein can be produced by genetic engineering techniques. Advances in genetic engineering techniques now permit the production of large quantities of almost any desired protein. In addition to making life much easier for biochemists interested in purifying specific proteins, this ability to churn out huge quantities of protein has given rise to an entire biotechnology industry. Figure 450. Companies now use bacteria, yeast, or cultured mammalian cells to mass produce all sorts of proteins that are used therapeutically, such as insulin, human growth hormone, and even the fertility-enhancing drugs used to boost egg production in women undergoing in vitro fertilization. Preparing these proteins previously required the collection and processing of vast amounts of tissue and other biological products, including, in the case of the fertility drugs, the urine of postmenopausal nuns. Using the same sorts of genetic engineering techniques, scientists can also design proteins that perform novel tasks, metabolizing toxic wastes, synthesizing life-saving drugs, or operating under conditions that would destroy most biological catalysts. We shall discuss these methods in great detail in Chapter 10. Automated studies of protein structure and function are increasing the pace of discovery. Biochemists have made enormous progress in understanding the structure and function of proteins over the past 150 years. See Table 4.2, page 162. These advances are the fruits of decades of painstaking research on isolated proteins, performed by individual scientists working tirelessly on single proteins or protein families, one by one, sometimes for their entire careers. But many future advances may come with proteomics, the large-scale study of cellular proteins in which the activities or structures of hundreds, even thousands of proteins are analyzed by highly sensitive, automated techniques. If scientists can perfect such methods, they might someday be able to monitor all of the proteins that are present in a cell, assessing whether they are switched on or off, and seeing which proteins they are partnered with, all in a single experiment. Large-scale analyses of protein structures are already underway. Techniques that have been miniaturized and automated allow researchers to rapidly clone genes, produce proteins, grow crystals, and collect X-ray diffraction data for hundreds of proteins at a time. Through X-ray crystallography and nuclear magnetic resonance (NMR) spectroscopy, see how we know, page 160, we now know the three-dimensional shapes of more than 20,000 proteins. These structures are archived in large, publicly available databases, Movie 413. By analyzing the conformations of these proteins, biologists have come to the conclusion that the vast majority of protein domains fold up into a limited number of patterns, perhaps as few as 2,000. The structures of about 800 of these protein folds have been determined so far. By studying how these patterns form, scientists hope to develop computational methods that will be able to take any amino acid sequence and predict both the structure and the function of the protein. Even with such information, it will still be an enormous challenge to decipher exactly how all these proteins, about 400 in the smallest bacterium and 10,000 in a typical human cell, work together to form a living cell. Figuring out exactly how proteins collaborate to create and maintain order in a universe that is always tending towards disorder will require both the continual development of new techniques and a great deal of human ingenuity. But the closer we get to answering this question, the closer we will be to understanding the fundamental basis for life. Essential concepts. Living cells contain an enormously diverse set of protein molecules, each made as a linear chain of amino acids covalently linked together. Each type of protein has a unique amino acid sequence that determines both its three-dimensional shape and its biological activity. The folded structure of a protein is stabilized by non-covalent interactions between different parts of the polypeptide chain. Hydrogen bonds between neighboring regions of the polypeptide backbone often give rise to regular folding patterns, known as alpha helices and beta sheets. The structure of many proteins can be subdivided into smaller globular regions of compact, three-dimensional structure, known as protein domains. The biological function of a protein depends on the detailed chemical properties of its surface and how it binds to other molecules, called ligands. When a protein catalyzes the formation or breakage of covalent bonds in a ligand, the protein is called an enzyme, and the ligand is called a substrate. At the active site of an enzyme, the amino acid side chains of the folded protein are precisely positioned so that they favor the formation of the high-energy transition states that the substrates must pass through to be converted to product. The three-dimensional structure of many proteins has evolved so that the binding of a small ligand can induce a significant change in protein shape. Most enzymes are allosteric proteins that can exist in two conformations that differ in catalytic activity, and the enzyme can be turned on or off by ligands that bind to a distinct regulatory site to stabilize either the active or the inactive conformation. The activities of most enzymes within the cell are strictly regulated. One of the most common forms of regulation is feedback inhibition, in which an enzyme early in a metabolic pathway is inhibited by its binding to one of the pathway's end products. Many thousands of proteins in a typical eukaryotic cell are regulated either by cycles of phosphorylation and dephosphorylation, or by the binding and hydrolysis of GTP by a GTP binding protein. The hydrolysis of ATP to ADP by motor proteins produces directed movement in the cell. Highly efficient protein machines are formed by assemblies of allosteric proteins in which conformational changes are coordinated to perform complex cellular functions. A regulatory protein code based on the covalent modification of multiple amino acid side chains allows each cell to control the location and assembly of its protein complexes. Starting from crude cell homogenates, individual proteins can be obtained in pure form by using a series of chromatography steps. Purification allows the detailed properties of a protein to be revealed by biochemical techniques and its exact three-dimensional structure to be determined.